Hi everyone, this is your instructor Christina Howard here, and this is the second part of the circulatory system presentation. So, without any further ado, let's get started. So where we ended last time was we were talking about complicated plaques in arteries. This one happens to be a coronary artery, which is one that supplies the wall of the myocardium itself. So these are really important because if they don't work properly, you get a myocardial infarction, aka a heart attack. So let's just add that. So, as we discussed last time, plaques are not as simple as cholesterol sticking to the walls of your artery. Instead, macrophages will bring triglycerides and other lipids from the surface of the arterial endothelium down underneath that endothelium and become what are called foam cells. And then that causes smooth muscle proliferation due to signaling and increased synthesis of scar tissue, essentially, around the plaque. So if your body can't get rid of it, one of the immune responses that it's kind of its backup or go-to is just basically the equivalent of, I don't know, throw a blanket on it, which is just surround it with collagen and hope for the best. Same deal applies to things like cysts. So cysts have a sac around them made of collagen, and that's the body's attempt to wall off something that it's incapable of dealing with. So that applies here as well. I'm trying to minimize the amount that I sniffle during this. I have some seasonal allergies going on, but it's a trade-off between positioning the mic close enough to my mouth that you can hear me, but then you also get to hear my nose breath. So, there's pros and cons to all of this. All right, moving on. Whoa. What happened there? All right, I want to move here. There we go. So, here we have a thrombus. So notice, same general idea is that the lumen of the artery is occluded, occluded meaning, of course, blocked in some way, but different in that this is a thrombus, meaning a clot, rather than a plaque. And you can also see that it sits on top of the tunica intima rather than being inside of it. So these are dangerous. Clots, well, older people are more prone to clotting. So this is why they wear things like compression socks, for example, to prevent blood from pooling in the extremity. But if blood does pool in the extremity, then you risk a thrombus forming, and then when blood pressure changes, you can have what's called a thrown clot, where the clot dislodges from its place of formation and then goes and travels somewhere else. So if you get a 90% or more reduction in blood flow, and this is again coronary artery, so supplying the myocardium, basically if it's reduced but not completely blocked, you get what is called an acute ischemic event. So this is another way to say this is sometimes termed a transient ischemic attack, or a TIA. And a lot of times TIAs are talked about on terms of stroke, but they are not limited to the brain. You can also have one that affects the function of the heart. So this is characterized by angina, which is chest pain. And that's because ischemia hurts. So it's painful when your tissues don't receive oxygen. If you've ever held your breath for too long, you know that. So this is associated with loss of oxygenated blood flow to the heart when it's supplied by the effective coronary vessel. So let's say this was in your left anterior descending coronary vessel. That would be very bad, obviously. So typically, if you get thrombosis, you get angina, and that usually precedes a myocardial infarct. So you'll get this angina pain as the clot is starting to build up and occlude the artery, and then once it's fully occluded, then you get a heart attack. So infarction is the basically medical term for lack of blood supply to the myocardium causes an area of the myocardium to die. And unfortunately, we don't have the ability to regenerate our cardiomyocytes as adult humans. So they don't come back, they get replaced with scar tissue. So a mural thrombus may develop. So intramural would be inside of the tunica intima. Mural is just on. So intra is within, mural is just on. And it often is associated with some kind of endothelial problem. So the endothelium of arteries and veins is designed to be smooth and facilitate what's called laminar flow. One of the processes that facilitates clotting in a healthy person is when a blood vessel is broken, sticky collagen fibers underneath the smooth part stick out, and that provides a place for platelets to stick and gather. If you don't have a broken blood vessel, but you do have dysfunctional or problematic endothelium, a clot or thrombus can generate spontaneously, and sometimes they happen to accumulate over pre-existing plaques because the area of the tunica intima associated with the plaque is damaging the endothelium in some way. So now we're going to jump off into blood and hemopoiesis. So hemopoiesis is the production of blood. And blood is super interesting because, it's that red goop that if you lose too much of it, you die. But it's more complicated than that. Blood serves a variety of purposes, including, but not limited to, gas exchange and nutrient delivery. And it has a whole bunch of other stuff as well. Excuse me. So instead of reading this slide to you, we're just going to dive right in. So this is what we're going to cover. So here's a picture you've seen before. This is the circulatory system, consisting of the heart, which is a muscular pump, blood vessels, and of course, blood. So we've talked about the pump, we've talked about the vessels, and now we're moving on to blood. And it feels appropriate to get a red pen now, so let's do that. I'm going to make it drippy just for fun. So, why have a circulatory system at all? Well, transport is one, and I mentioned that. So basically, you need to deliver gases and take wastes away from your tissues as they accumulate. And then hormones and stem cells are also very important. The idea with transport, especially as it relates to blood, is that it takes too long to rely on diffusion for this. So, for example, diffusion rates determined by the Fick equation, which we won't get into here. Basically, one of the components or mathematical components of that calculation is distance. So the larger the diffusion distance, the smaller the rate of diffusion. So if you think about getting some oxygen from your lungs, where you take it in, all the way down to your feet, that's quite a long diffusion distance. And in fact, it would take, I think I calculated something like 27 years or something like that for that to happen under diffusion. So if you wanted to diffuse something to your feet from your lungs. So instead, you've got a pump and you've got fluid that is moving through and being moved by that pump. Protection is another one. So we have white blood cells, and these are able to travel in the blood to a variety of locations. And they can take care of infections, they can destroy microbes. They can even go so far as to neutralize cancer cells or precancerous cells. We'll talk more about that later. And then fluid balance, pH balance, and temperature control is another important one. So there's all kinds of reasons to have blood and also to move that blood around. So the circulatory system is heart, blood vessels, and the blood. The cardiovascular system is only the heart and blood vessels. And then if you want to be a hematologist, that means you're specializing in blood and blood only. Okay, so components and general properties of blood. So this is an image that's just showing the withdrawal of blood from somebody's arm and what happens when you put it in a centrifuge. So as adults, we all have between 4 and 6 liters of blood. And of course, that's going to be proportional to body size, right? So somebody the size of like Andre the Giant is going to have a lot more blood than a very, very small person. Obviously, bigger person, more tubing, more blood needed. Blood is considered to be a connective tissue. And I'm happy to report that many of you got that correct on the exam. So I said, which of the following do vampires drink? And you said a connective tissue. So you successfully made that leap. Very good. So when you spin blood down, what happens is the denser parts, which we call the formed elements, precipitate out and settle at the bottom. So plasma is the matrix of blood, the extracellular matrix. And it is a fluid matrix. So this is a light yellow fluid consisting mostly of suspended proteins and ions. The formed elements include red blood cells, also called erythrocytes, and then less than 1% of that is either white blood cells or platelets. So less than 1% of whole blood is your immune defense system. You can estimate oxygen carrying capacity using hematocrit, which is packed cell volume. So this is the percentage of whole blood which is comprised of red blood cells. It tends to be slightly higher in XY folks than in XX folks, just because typically folks with the Y chromosome produce androgens, and that increases hemopoiesis a little bit, but not always. Of course, there's a huge variation in the population. That's just if you average like a whole lot of people, you get this slightly higher hematocrit on average. So if you go to the hospital, for example, your hematocrit may be measured with what's called a pulse oximeter. And that is the little clampy thing with the red light that sticks on your finger. Basically, it's shining red light through your finger and seeing what percentage of that red light is reflected back and what makes it through. And that's an indicator of redness, which is an indirect indicator of how many red blood cells you have. So that's what that does. But the best way to actually and accurately measure hematocrit is, of course, take blood, use a centrifuge. Okay, so there are 7 kinds of formed elements, and these are erythrocytes, eryth meaning red, platelets, which are actually not cells, they are fragments of cells. They come from a special cell in the bone marrow called a megakaryocyte. So let's say fragments of megakaryocyte. So you may see platelets referred to as thrombocytes. Thrombus is a clot, and oocyte means cell. That's not quite accurate because they're not cells, but that's a name that exists, so I want you to be aware of it. Leukocytes, or white blood cells, you have different types of these. So think of leukocytes as being a family of cells, including the cells that are called granulocytes because their cytoplasm has granules in it, so neutrophils, eosinophils, and basophils, and agranulocytes, so ones without cytoplasmic granules, and those are lymphocytes and monocytes. So remember from the previous slide that less than 1% of blood is made up of these cell types. Of these cell types, or within the leukocyte group, there is differences in relative abundance of leukocytes. So this will help you when you're looking for these cells in lab. It's just helpful to remember this little mnemonic for the relative abundance of leukocytes. So I'm going to make a little space for myself to write here just by scribbling, and then I'll go back to this. So the mnemonic device for remembering the relative abundance of leukocytes from most to least is as follows. Never let monkeys eat bananas. That's pretty easy to remember. Never stands for neutrophils. Let stands for lymphocytes. Monkey stands for monocytes. Eat stands for eosinophils. And bananas stands for basophils. So The reason I'm mentioning this is because as you look for these cell types in your virtual slides, you're going to have to spend a lot of time looking before you find a basophil, because white blood cells are less than 1% of whole blood. Basophils are less than 1% of that, less than 1%. So they're very rare. But they do exist, and I'll show them to you. So that's the relative abundance. So you do need to be able to tell me which of these are the granulocytes versus agranulocytes. And by the end of this, also tell me the general function of each of these kinds of cells. So here's that breakdown once again, just to show you relative abundance. And this also includes about how many cells per cubic millimeter of blood there is. It also breaks down the contents of plasma very effectively. It's something I like about this chart. Okay, so moving right along, let's talk about plasma. So plasma is just the liquid portion of blood, and it's not the same thing as serum. So serum is what happens when you separate the plasma from the whole blood, and then you spin the plasma down, which means that you're going to take any suspended solids and clots and proteins and remove those. So serum is very similar to plasma, except for it lacks the clotting precursor fibrinogen. We'll talk about fibrinogen as well. It's a liver protein that is a precursor for clotting. So 3 major categories of plasma proteins. Albumins, which are a group of proteins. These are the smallest and most abundant, but they make the largest contribution to viscosity and to osmolarity. So the amount of albumin in one's blood has a direct consequence on osmotic pressure of the blood, so how much water is gained and lost from the blood at the capillary bed, and therefore fluid balance, so how much water is outside your cells versus how much water is inside of your cells. Globulins are also called antibodies, and these are non-living proteins that have a kind of a Y shape in general. And these are going to stick to pathogens that the immune system remembers. And then fibrinogen is a precursor of fibrin. So basically it's an inactive clotting agent. When it's activated, it deposits out of the plasma and forms a clot. Okay, so this is just showing you how a blood slide is prepared. So you are going to prick your finger and take a small amount of blood, and then using a micropipette and capillary action, draw the blood up the pipette and then blow it out onto a slide. Then you perform what's called a smear, where you put the edge of another slide on and then drag it across in the direction shown. So what this is going to do is create a quote-unquote lawn of spread out blood cells so that you can more easily observe them. The goal is to get them to a state that looks like this, where the cells are dispersed enough that you can see them being separate. Oopsie. Okay, so let's talk about the first cell type, and these are erythrocytes. So erythrocytes are very important. They have 2 principal functions, and those functions are to, 1, carry oxygen from the lungs to the cells, and 2, to pick up carbon dioxide, which is a waste product of oxidative phosphorylation, and bring that back to the lungs. So because your tissues rely so heavily on being oxygenated, if you don't have enough oxygen carriers, which is what erythrocytes are for, things get very, very bad very, very fast. So severe blood loss due to hemorrhage is likely to send you into shock and subsequent death relatively quickly. Fortunately, you have systems in place to prevent that from happening, so we will talk about the renin-angiotensin system when we get to the endocrine system a little bit. But yeah, so just in general, avoid bleeding. I think that's obvious to everyone, right? Okay, so we have these little discoid guys, and as you can see from this SEM, and it's a false color SEM, so this image would be gray if somebody hadn't bothered to put colors on it, but you can see that they are shaped kind of like an almost donut. So they have this nearly toroid shape, almost donut, such that they're flattened in the middle. That's what this middle portion shows you. And really what's in here is mostly just hemoglobin. So during the development of erythrocytes, they used to have a nucleus and organelles. Once they differentiate sufficiently, they will spit those out and flatten and become a fully mature erythrocyte. So your tiniest blood vessels, your tiniest capillaries, are so little that sometimes red blood cells have to fold in half like a taco to get through them. So that's one of the significances of this shape, is that it's easily foldable, unlike a sphere. The other is that they can flow through tissues in these stack-like arrays called a rouleau, which is just a French word. So that's what that means. It's a fancy way of saying they proceed through the blood vessel single file. So, they're about 7 microns in diameter. This is a good indicator of their size, and actually you can use them as a benchmark to estimate the size of leukocytes when you're using the microscope. So they have that diameter, and they run from about 2.6 microns at their widest to 0.8 microns at their thinnest. So when you look at them on the microscope slide, the middles look almost transparent, actually. So during development, they used to, like I said, have organelles and a nucleus. They don't anymore once they're mature. So they lose all of them during development. So a young, early erythrocyte and you can see some of these circulating actually on slides. It's called a reticulocyte. And reticulocyte refers to the reticular network of decaying membranes, primarily RER, as the cell undergoes the process of autophagy and eats up its own organelles. So that's the deal there. They also don't have mitochondria, and that's for a good reason. So mitochondria use oxidative phosphorylation to produce ATP, right? That requires oxygen. If the whole purpose of this cell is to deliver oxygen to cells and tissues, one thing that you don't want it to be doing is eating up the oxygen on the way to those cells and tissues. So instead, these use anaerobic fermentation, aka glycolysis, to produce ATP. Oh, if you heard that chirp in the background, I apologize. My bird got out of his cage and is loose in my office, and I can't catch him. So he's my temporary office mate until I figure out a way to get him back into his cage. He's a tiny criminal. So red blood cells, therefore, don't have a nucleus, they don't have any DNA, and they also don't do any protein synthesis or mitosis. So they're functionally in G0, they are amitotic. This is why you must constantly be producing them. And that's where the kidneys and erythropoietin come in. So there is a disease called aplastic anemia. Aplastic means not making. So basically what it means is you are having anemia due to not making enough red blood cells. We'll continue to build up this concept. Okay, so a little bit more about erythrocyte morphology. You can see the rouleau on the right-hand side here, and then you can see a venule and the erythrocytes passing through it on the left. So I mentioned aplastic anemia. In general, anemia is just you have not enough erythrocytes. You can also have iron deficiency anemia, where the erythrocytes that you do have just don't have enough hemoglobin in them, or you can just have straight up not enough of them. So this is a problem because it subjects the individual to long-term hypoxia, which is lower than normal oxygen, and that basically makes you feel crappy. So your skin gets pale, you're tired all the time, you get heart palpitations, and that can happen due to a variety of reasons, including, but not limited to, vitamin B12 deficiency. So quick shout out to vitamin B12. If you don't take it, consider doing so. here's the deal with vitamin B12. So if you want to absorb dietary iron, you have to complex it with B12. And unfortunately, you don't really make your own vitamin B12. You have to get it from other sources. So if your diet is poor in B12, you may suffer from anemia due to an inability to absorb iron. So you could be eating plenty of iron dietarily, but if you don't have its little friend, B12, you won't get any benefit from it. Most folks who consume animal protein aren't really at risk of this because there's plenty of vitamin 12 in animal protein. Other animals concentrate it in their tissues and then we eat them. However, folks on, for example, a entirely plant-based diet might consider supplementing with B12 in order to facilitate iron absorption. Unfortunately, there's plenty of vegan sources of B12, including but not limited to delicious, delicious nutritional yeast or Marmite, if you're that kind of person. I am. So make sure that you're getting your B12. Okay, the opposite is also a problem. I know, right? Like, you can't win. You can either have too few red blood cells or too much is also a problem. You need the Goldilocks amount of red blood cells, but it's true. So polycythemia is also called erythrocytosis, and this is just lots and lots of red blood cells. So a higher hematocrit per unit plasma than ordinary people. Now, in some cases, it can be an adaptation to living at high altitudes. So, for example, there's that classic example, the Andes, Machu Picchu. Guess what? I don't know how to spell Machu Picchu. I just found that out about myself. Learn something new every day. I guess I'll go look. Regardless, these are really high altitude locations where people do live year-round and full-time. And these people probably have polycythemia as an adaptation to the low O2. So if the oxygen ambiently is low, better have more ways to grab it out of the air, right? However, elevated hematocrit makes your blood goopier. It's slower to flow, there's more resistance, and increase in resistance puts strain on the heart. And that can impair circulation through the capillaries if it's extreme, but long-term, the strain on the heart is the real problem because, of course, it can cause hypertrophic left ventricle, which you guys saw previously. So although polycythemia can be a physiologic adaptation, humans, for most part, aren't really designed for it long term. Okay, so let's talk about hemoglobin. So before your erythrocytes lose their nucleus, the endomembrane system in the nucleus of your erythrocytes, your immature ones, are hard at work making hemoglobin. So hemoglobin has these protein chains, alpha and beta chains, and those are the globins. So let's underline that. Those are these guys, beta and alpha. So that makes hemoglobin an tetramer, meaning 4 subunits, 4 proteinaceous subunits. Inside each of those globin chains is a heme group, and the heme groups are basically a flower-shaped array. They're a pigment with ferrous iron at the center. That's Fe2+. Ferric iron is Fe3+, not the kind we want. Ferrous iron is Fe2+. So, in this configuration, the hemoglobin molecule appears red. It reflects red light, and that is why your blood is red. Fun fact, not all organisms use a red pigment in their blood. Some organisms use other pigments. So, for example, horseshoe crabs have blood that is blue instead. It just depends on what pigment you're using to grab onto gas. So, red blood cell count and hemoglobin concentration can both be used as an indirect measurement of how much oxygen your blood can carry. So that's typically assessed using hematocrit, which is the percentage of whole blood composed of red blood cells, but you can also use a pulse oximeter to describe hemoglobin concentration, because pulse oxes only measure redness. So you could have really red blood either because you have extra good hemoglobin or because you have a lot of red blood cells, usually the latter. Okay, so one thing to note is that adults have mostly alpha and beta chains of hemoglobin. Everybody had a gamma chain when they were a fetus. The significance of that is that this particular globin has a higher oxygen affinity, which assists with fetuses taking oxygen from their mom's blood across the placenta. So it's an easy way to think about that is their hemoglobin is just ever so slightly grabbier of oxygen than adults. So it's a little bit easier for oxygen to hop off of mom's blood and hop onto babies. And then so for most of your life you have mostly alpha and beta chain hemoglobin, but less than 3% is this other kind, the delta chain. This one is hemoglobin H disease, and I'm actually not super familiar with this disease. It looks like, I mean, obviously it's one that's comprised only of beta chains, so maybe the genes for alpha are defective in some way, so you only make beta, but I'm not sure what consequence that has on physiology. I'll have to look it up. This is why I like my job. I get to learn new things. And then slowly become more smart and more powerful over the years. Okay, so interestingly, hemoglobin has an impact on the structure of red blood cells. So that little toroid shape that they are, toroid means almost a torus. So a torus is a donut. Toroid means nearly a donut, right? So it's pinched in the middle, not hollow. So hemoglobin is responsible, in part, for the toroid shape of erythrocytes. If you have a single nucleotide substitution, so just one A, C, T, or G in the hemoglobin gene, that can change the sequence of amino acids, so it's not a silent mutation. And that produces a version of hemoglobin that is prone to polymerizing, so making clumps of itself or chains of itself. And that change actually ends up changing the cell shape and also reducing their flexibility, so they can't do that taco thing. So if you have somebody who's homozygous for this mutation, this can lead to basically sludgy blood, and because they can't taco through the small vessels, microvascular disease and circulation. So basically you get pileups of these sickle-shaped cells at the entrances to small vessels, and that causes ischemic bouts until the blockage is cleared, that are very painful for the people that suffer from them and that are often dangerous. Okay, I think I'm going to stop here for this particular video because it's gone on long enough, and also I don't want to start leukocytes and not finish them. So the next video is going to concern leukocytes and what their jobs are and why they're so diverse, and we'll jump off from there into the lymphatic system and immunity, which is especially relevant right now in the times of COVID. So I'm looking forward to helping you guys understand viral biology a little bit better and helping you understand fighting off viruses a little better in the next few lectures. So thank you for your attention, and I will see you next time.